So networks of spiking neurons are increasingly used in neuroscience to understand mechanisms underlying phenomena observed in electrophysiological recordings in vivo. And there is a long list of questions that people have addressed, and I will mention a few of them during my lecture. For instance, the irregularity of firing that is ubiquitous in cerebral cortex in vivo, broad distribution of firing rates, correlation structures, the emergence of synchronized oscillations, and many others. So to study networks of spiking neurons, there are two possible strategies that I will describe briefly. The first strategy, the most straightforward, is to use numerical simulations. And you will have later today a tutorial exactly on this strategy. So how to simulate networks of spiking neurons using in particular the software NEST. Another possible strategy is to use analytical methods, in particular methods from Meanfield theory that I will mention briefly later in this presentation. So what are the pros and cons of both methods? So numerical simulations, the advantage of simulations is you can use them to simulate any network model you like. Of course, you will be typically limited in the number of neurons that you can simulate. But the advantage is that there is no need to make any approximations. You can implement pretty much anything. And nowadays, it's possible to simulate very large networks. You can easily simulate networks of tens or hundreds of, even hundreds of thousands of neurons, depending, of course, on the complexity of single neurons, and depending on what kind of machines you is available. However, there are also drawbacks. One major drawback is that in many network models, you have a really high dimensional parameter space. So it's really hard to explore systematically. And it's hard to characterize parameter regions in which we find specific behaviors of interest. And so that's where analytical calculations are useful. The goal there is to really obtain a deeper insight into mechanisms underlying specific behaviors and to really find what are the critical parameters that control specific behaviors of interest. And I will mention a few examples in this lecture today. Of course, the drawback is that there are basically very few analytical solvable models in computational neuroscience, especially at the level of networks. So typically one needs to make approximations. And I will mention one example in my lecture today. And so ideally, one should combine both. And a typical strategy is to start with exploratory simulations of a particular model. And that's useful to understand the types of approximations that are likely to be successful in describing the system. Then you can go to the blackboard and try to study the simplified system analytically to get deeper understanding. And then maybe go back to simulations to check the validity of approximations. So there is potentially a fruitful dialogue between simulations and calculations. And of course, there is another dialogue that one always should have between your model and experimental data, which is another, of course, very important dialogue in computational neuroscience. All right, so how to build a network model? So basically, one has to answer the following list of questions to build a network model. And I will go through these questions one after the other in the next slides. And I will present in particular one particular model that is a randomly connected network of excitatory and inhibitory neurons, which in fact, that will be the example network that we will simulate later today. So the first three questions here, how many types of neurons, how many neurons, how are they connected, will specify the architecture of the network. And then the next three questions about external inputs, neuronal model, synaptic model, will specify the details of the basic elements of the network. So what about the first questions? How many neurons? So there have been, of course, the answers to all these questions depend on exactly the problem that you're interested in, the particular neuronal structure you're interested in modeling. For example, if you're interested in in a network model of the striatum, then you might want to include medium spiny neurons, maybe 2 classes of neurons, and maybe you want to include some interneurons, or fast spiking interneurons, cholinergic interneurons. And so the answer here will be basically specific to that particular structure. If you want to model the cerebellum, then maybe you want to include granule cells, Golgi cells, Purkinje cells, and so on and so forth. So for networks of cortex, a very useful model has been to include just 2 populations of neurons. One population is the population of pyramidal cells, excitatory neurons, and the other are inhibitory interneurons. But of course, people have also studied a more detailed model where, for instance, one includes more inhibitory subtypes. A popular model nowadays has 3 different inhibitory subpopulations, and also extensions have included models where you have multiple layers, or you can also include models at a larger scale where there are multiple areas or structures. So in this lecture, I will mention a network where you have a large number of neurons, and 80% of these neurons are excitatory, 20% are inhibitory. These are the typical fractions of neurons that one observes in cortex. The next question is, how are these neurons connected together? And again, there are many possible answers to that question. I will be focusing in my lecture on a very simple connectivity structure, which is a purely random connectivity. So in other words, the network will be an Erdős-Renyi random graph. Since there are 2 populations of neurons, the connectivity matrix will have a block structure, 2 by 2 block structure, which is indicated here. So for instance, for excitatory to excitatory connections, existing connections will have a synaptic weight JEE, but then the existence of a connection, Cij, will be a random variable, will be a Bernoulli variable. It will be equal to 1 with some probability C. C is the connection probability, and 0 otherwise. And we see that this random variable is present in all blocks. For simplicity, the connection probability will be the same for all types of connections, but the connection weight will depend on the type of connection. For example, you have a connection weight minus GEI for inhibitory synapses onto excitatory neurons. So in this network, each neuron will receive a number of connections, an average number of connections per neuron, which is KE, which is basically the product of the connection probability times NE. This is the average number of excitatory connections, and then an average number of inhibitory connections, KI, which is equal to C times NI. And typically, we will investigate this network in a regime in which both numbers are large. KE and KI will be of order hundreds or thousands, because this is what the anatomy tells us, in particular in cerebral cortex. So now let's specify the external inputs. So we'll assume that this network receives random external inputs from outside the network, and we'll describe them as independent Poisson processes. So that will lead to synaptic currents to each neuron in the network IIX, which will have this form. JAX will be the amplitude of individual postsynaptic potentials triggered by external spikes, and then spikes are modeled as delta functions. Right, they are instantaneous. They are instantaneous in time, and TJK here is the kth spike of neuron J, and these spikes are generated according to independent Poisson processes. And the motivation here is that in cortex, the spike trains of neurons recorded in cortex are highly irregular, and the spike trains bear a lot of similarities with Poisson processes. So what about single neurons? So there are a huge list of potential single neuron models that one can study. At the network level, the leaky integrate and fire neurons have been very popular because they are basically essentially the simplest possible spiking neuron models that you can implement. They are described by a membrane potential variable that evolves according to the simple first order differential equation. Here tau I is the membrane time constant. You have here a leak term, so that the voltage goes to a resting membrane potential in the absence of inputs. But then in a network, these neurons will be driven by external inputs here, these IIX that I introduced, and then the recurrent synaptic inputs coming from all the other neurons in the network that are basically that depend on the connectivity matrix that I have introduced.
Nicolas, can I interrupt you for a second?
Yes.
And so in the previous slide, in the definition of IIX,
yes,
you also had a tau I.
Yeah, this is just for dimensional reasons. So delta
okay. So you
one of the time units.
Yeah.
So
yeah.
basically, in this case, this is in millivolt units, and J is in millivolts. This is just a convention. I mean, I could absorb it in the J's, but
okay.
Okay, so the last thing to specify is this SJs. So S basically is are the postsynaptic currents induced by single presynaptic neurons. Here's a presynaptic neuron J. Again, there are many possible descriptions for synaptic currents, and here I will essentially focus on the simplest possible description for a network of spiking neurons, which is to take again delta functions for synaptic currents. So the only parameter describing the synaptic currents is a delay. So this indicates schematically a spike train of a presynaptic neuron, and then the postsynaptic neuron basically receive this action potential with a delay T. With these postsynaptic currents, the membrane voltage will make a jump of amplitudes Jij whenever it receives a spike from neuron J, and then it will decay exponentially with time constant tau I in between action potentials. Okay, so these equations specify basically everything about the network. Then you have to choose initial conditions, and then you can simulate it using these equations. So now what kinds of behaviors do we see in these networks when we start to explore the dynamics? So in a broad range of parameters, and that mimic the parameters that can be estimated from anatomy or electrophysiology, we see that these networks settle in so-called asynchronous irregular states. And these are illustrated here in this slide, which is taken from a review paper by Tim Vogels et al. So this represents the spike times of a small fraction of the neurons that are present in the network. Here is a simulation of the network of a few thousand neurons. So to be able to see the spike times, you just have to select a small fraction of them. And you see that these spike times are very, very much random looking. In fact, if we zoom on one of these neurons, we see that we have large fluctuations in the membrane potential below threshold. And then occasionally, the membrane potential will hit threshold and a spike will be generated and will be communicated to all other neurons in the network. Because the network is active, in fact, the neurons receive a large bombardment of inputs from other neurons. So this leads to large excitatory currents here in green and large inhibitory currents in red. And interestingly, these two currents approximately cancel each other. So the sum of these currents here is much smaller than the individual components, excitatory or inhibitory currents. And so that leads to something that has been termed the balanced state. And so this irregular firing can be quantified by distributions of interspike intervals, which show which are very broad and typically have an exponential tail, and the coefficient of variation of the interspike intervals, which is the standard deviation of this ISI distribution divided by the mean, and it's typically of order 1, so consistent with similar to a Poisson process. Another feature of these networks is that typically the distribution of average rates is very broad. As you can see here, that can be seen from these raster plots. Some neurons, you can see some horizontal lines that have many more spikes than others. And this is true even though the neurons are all identical in this network. So the only thing that generates this broad distribution of firing rates is the random connectivity. All right.
Nicolas, can I ask another question?
Yes, sure.
So the neurons that in this case have higher firing rates, they also have lower CV or is that, I mean, is there a systematic relationship between the?
Yes, very good question. Yeah, yes, there is. Typically, but not always, the CV is a decreasing function of firing rate, though sometimes it can be non-monotonic. Sometimes, so that's where calculations are useful. So if you make an approximation, Gaussian white noise approximations for synaptic inputs, then you can show that in the low rate limit in leak integrate and fire neurons, the CV has to go to 1 in the low rate limit. Okay, so when neurons fire at very low rates, then the CV has to be close to 1. But then it can either, when you increase the firing rate, for instance, by increasing the external inputs, then it either decays monotonically. And so in fact, when neurons fire at very high rates, you can show that the CVs has to go to 0, because when they fire close to saturation, the firing has to be very regular. But it can also be non-monotonic. Sometimes you can have supra-Poissonian behavior. And that depends a lot on the reset. If you reset close to threshold, then you will have, for some range of rates, a bursty behavior where the neurons tend to fire a burst of spikes.
But also in the same network, without changing the external input, that kind of non-trivial relationships exist, right?
Yes, right. Yeah, exactly. All right. So now, as I said at the beginning, so you can simulate these networks, but you also try to analyze it mathematically to try to understand what goes on. And so let's have let's have a closer look on the synaptic inputs to neurons in this network. So here I'm just rewriting the total inputs to a particular neuron I, and this is basically the same equations that I write in one of the first slides, except that now you can see more clearly that there are external inputs, excitatory and inhibitory inputs to this neuron. So the idea is that we want to look at the statistics of these synaptic inputs, because basically the external inputs are basically Poisson processes. We will approximate the spike trains of presynaptic neurons also as Poisson processes, even though they are not exactly Poisson processes, right? Except, as I just said in answer to Yasser's question, in the low rate limit. And what we'll see is that we can divide this in practice these inputs in three parts. So mu e is the average, are the temporally average synaptic inputs, but also average over all neurons in the network, right? But as we will see, there are fluctuations in the average inputs from neuron to neuron due to the randomness of the synaptic connectivity. So there is this delta mu I, which will tell us how the mean input of this particular, where the mean input of that particular neuron is. It can be higher than the global average, or it can be lower. And then there are the temporal fluctuations due to the fact that spikes arrive at random times. They are because of this irregularity of firing. And in fact, we can easily estimate the mean inputs. The mean inputs mu e will be just a product of the total number of connections times the individual synaptic strength, so JEX for external input, JE for recurrent recurrent excitatory inputs, and G minus GEI for inhibitory inputs, and then times the firing rates of these different types of corresponding to these different types of inputs. So that's simple. Then you can also compute the fluctuations in mean inputs from neuron to neuron, and you can compute the variance. And this is this calculation is also can also be done. And you see that it will be also proportional to k, but now it should be proportional to j square, to the square of the weights and to the square of the firing rates. Now, to compute the temporal fluctuations due to spiking activity, this is a little bit more tricky, and that's where you need to make an approximation, because so you need to make an approximation. In particular, you need to approximate the spike train as Poisson processes, which, as I said, they are not exactly Poisson processes, and then you have to neglect the fluctuations you have to neglect, sorry, the correlations between spike trains. And that's where, basically you have to assume that the network state is asynchronous, but that's an assumption that can be checked later. In this case, you can compute the variance of these temporal fluctuations. And you see that, again, they will be proportional to k and to the square of the synaptic rates. All right, so the idea is that now we can make a diffusion approximation. So we have a complicated stochastic process here because these spikes arrive at discrete points in time. So now we make an approximation that this is we approximated by a Gaussian white noise with the same statistics as the original process. And when you do this approximation, basically what that allows you to do is actually to compute the firing rate. You can compute the firing rate of a neuron receiving this kind of inputs. And basically what you get in this way is you get a transfer function that as a function of the mean input of a particular neuron and its fluctuation, sigma e, you get its firing rate. And in fact, this can be done in a few models, in particular the leaky integrated fire models, but in fact, also other nonlinear one variable integrated fire models. And it can be either approximated or computed numerically for other models. And that allows you to compute self-consistently the firing rates, the average rates in the network. Basically, what you're doing now, the next step is that this is essentially a Gaussian random variable in the large N limit. And so to compute the mean rate, so the rates are different from neuron to neuron, but essentially this is a Gaussian random variable, so you can compute the mean rate using this Gaussian integral, basically. And you can also compute the distribution of rates in the same way. And that's something that you can compare with the simulations that show. All right. So that's a way to get analytically the mean rates in these networks. So now we'd like to go further, and we'd like to understand the behavior of these equations in specific limits. And in particular, it's tempting to use to look at the limit when k is large, is very large, because as I said, the k's typically in cortex are on the order of several hundreds of thousands. So it makes sense to look at what happens in that limit. Then you realize that when you take this limit, you have to choose a particular scaling for the J's, for the synaptic couplings. And the reason is that if you just let k go to infinity, all these quantities will diverge, right? The mu's, the variance of delta mu and sigmas. So when you look at this equation, it seems like the scaling that makes sense would be 1 over k, because if the J's scale as 1 over k, then the mu's will go to a finite limit in the large k limit. However, the problem there is that in that limit, then, if J scales of 1 over k, you can see that both the variance of delta mu and the variance of the temporal fluctuation, sigma, will go to 0 in that limit. And so that will be at odds with what you see in that picture, where you have very large fluctuations of both inputs and consequently membrane potential. So that won't give you a good description at all of what happens in these networks. And in fact, that's also consistent with the fact that estimates of J's in cortex, when you estimate k and j, basically the J's are much larger than 1 over k. Let's say k equal 1,000 would mean that J's are of order 10 to the minus 3 times the threshold, and the PSP amplitude in cortex are much larger than that. So the next scaling we can try is 1 over square root of k for J's. And the reason then for 1 over square root of k is that we can see that this is the only scaling that will leave both the variance of delta mu and the variance of temporal fluctuations finite in the large k limit. Okay. So that's the right limit if you want to investigate a network state that is fluctuation driven, where fluctuations remain finite in the large k limit. However, now it seems like you have a problem, because now the mean inputs, now it looks like they will diverge in the large k limit, right? So you have to do, and that it looks bad, because divergence of mean inputs mean either you go, neurons fire at saturation or they're completely silent, right? Depending on the sign of this term. But actually, this term can, what happened is that this term actually cancel approximately. In fact, they cancel up to 1 terms of order 1 over square root of k, provided specific conditions on the connectivity matrix are satisfied. In particular, inhibition should be strong enough. The intuition is that inhibition should be strong enough to cancel the the large mean excitatory currents that neurons receive. So that leads to this kind of two sets of linear equations. So if we go in the large K limit, this will go to 0, the set of two linear equations for the firing rates in the network. Okay. And that explains this balanced state idea, the fact that excitation and inhibition approximately cancel each other in these networks. As a result, the mean input currents are below threshold because of this cancellation, and then the neurons fire due to fluctuations in their inputs. And so basically, the fact that sigma is finite, the variance of temporal fluctuations, explain the irregular firing of neurons, so the fact that they have a CV close to 1, and the fact that the variance of delta mu is finite, the fact that you have a finite finite fluctuations in the mean inputs to neurons, that explain why you get automatically a broad distribution of firing rates. While you would think naively that in the large K limit, basically these fluctuations would disappear. And that's not true when you use this K. All right, and in fact, these networks have been, because of these two features, these networks can reproduce quite successfully experimental observations in cortex. And so in particular, the highly irregular activity that you see pretty much anywhere you record from in cortex. In particular, these are recordings, intracellular recordings in vivo. This is a neuron that is recorded in an awake rat, and you see these very large fluctuations of memory potentials and the consequent highly irregular firing. And also this phenomenon of this very broad distribution of firing rates is also something that is seen pretty much everywhere one records in cortex. These are a few examples from 3 different species. And these, in fact, these distributions have been fitted pretty well by log normal distributions, in particular by this group, Tony Zader's group. And in fact, we can show that in some specific limits in networks of leaky integrated fire neurons, the distributions will also become closer and closer to a log normal. So we can make really a very quantitative comparison here between data and the model.
Nicolas, just one quick question. Yes. So you showed us the wonderful hallmark here of for balanced state, and you showed us that these terms cancel. So what would you need to do to put yourself outside this balanced regime?
Mm-hmm. Yeah, actually, that's my next slide. So
lovely.
Well, I mean, part of my next slide. I think there is a more complicated answer but to this, but let me describe this slide and then come back to this question. All right, so, okay, so what I describe is actually only one possible state that this kind of network can be in, which is so-called asynchronous irregular state. So asynchronous because you have very weak correlations between the firing of neurons. You can think about neurons as different neurons in the network as receiving essentially uncorrelated Gaussian white noises. So neurons become essentially uncorrelated Ornstein-Uhlenbeck processes, and then they each fire their own spikes depending on their own fluctuations. But however, this state is not necessarily, so first, this state, there are some conditions on synaptic connectivity for this state to exist. And by the way, so this phase diagram here was derived now, it's no longer in the large K limit. This is derived for a particular value of K and J. And basically what you see here, what you see clearly is that you need inhibition to be strong enough for this state to exist because it only appears in the right part of this plot. So basically when the IPSP strength, the inhibitory postsynaptic potentials seem to be large enough so as to overcome excitation. Otherwise, what happens is that you only have, the only possible network states are network states with very high activity, which might bear similarity with maybe epileptic states. And they also, to arise, you need also, there is a range of external inputs in which they arise. But now there are other things that can happen. And what can happen is that this state can destabilize due to various instability mechanisms. And you can see here that this state is surrounded basically by 3 types of synchronized states, where the neuron now will become highly synchronized. So this one is maybe the most boring. So that's when excitation starts to dominate over inhibition. So inhibition can no longer control the activity in the excitatory network. And depending on the details of synaptic transmission, what happens is that this state can become highly synchronized, as you can see here. On the other hand, here, there is another state in which, another synchronized state, which is perhaps more interesting, in which neurons are now, you see that the average activity in the whole network is no longer close to being constant in time, as in the slide I showed you. So I showed you previously. Maybe I can come back to that. Maybe I can come back to that slide here. We see that the population activity was basically fairly flat. So in this case, we see that we have pronounced oscillations of the population activity. And interestingly, so you have an oscillatory population activity, but single neurons are still firing quite irregularly, even though there is a pronounced population oscillations. And in particular, the population oscillation is at a really high rate in this case. This is something like more than 100 Hertz oscillation, but single neurons fire at lower, their firing rates are lower. So the idea is that in this type of oscillation, you have at each cycle of the oscillation, you have only a fraction of neurons that make it to threshold because of these temporal fluctuations in their synaptic inputs. So some neurons basically won't make it at a particular cycle, and maybe not at the next cycle, but maybe the following cycle they will make it, and then the spike is emitted. And so that can be called a synchronous irregular oscillation. It's been also called stochastic oscillations. And these actually bear a lot of similarities with oscillations that are seen in cortex or hippocampus, where typically, so in particular fast oscillations, so either the gamma range or the very fast oscillations seen in sharp waves, for instance, sharp wave ripples in hippocampus, typically single neurons fire at much lower frequencies than the population. And the mechanism here is a purely synaptic mechanism, and it's inhibition-driven oscillation, where when you have a peak of activity at a given point in time, then what happens is that because of the delay in synaptic transmission, neurons will receive a big surge of inhibition sometime later this peak, so their activity has to go down. Now, when you are at the trough of at the next trough, what happens is that then you will have much less inhibition coming in later, and so that allows neurons to come back up. And so you have this oscillation whose frequency depends primarily on synaptic time constants, and that can be analyzed in more detailed models in which you have more realistic synaptic time constants, description of synaptic activity. And you have also other types of oscillatory states that emerge. For example, this slower oscillatory state in which you have a burst of activity that alternate with silent periods.
Nicolas, I have a question.
Yes, sure.
So this distinction between slow and fast oscillation, is it a distinction based on just the frequency, or the mechanisms are different?
Yeah, the mechanisms are actually different, and they are, so here this is really a network which is, so the instability really relies on the properties of synapses. And so you have this very fast rhythm that emerge because, so it's basically a rhythm that's where you have this fast negative feedback into inhibition plus a delay, and that's basically delayed inhibitory feedback is a very good way of generating oscillations, as engineers know. This is a different mechanism. Here, this is a region where you have basically a bistable region between a silent state and an active state. And then the idea is that you have a slow oscillation between an active state that is then destabilized and go to the silent state, and then basically you have you have this alternation between active and silent states. So and here the oscillation frequency does no longer depend on the synaptic time constant, but rather depends on the membrane time constants and the external inputs also. So so I was describing the different types of mechanisms to generate network oscillations. So there is the purely inhibitory mechanisms, the E to I, EI feedback loop mechanism that typically generates a lower frequency range, maybe beta gamma frequencies, and that could be, for example, responsible for oscillations in sensory systems. And then you have networks that are you have other mechanisms that rely also on intrinsic or on slower intrinsic or synaptic time constants, for example, adaptation, short-term depression, and things like that. And that can lead to additional mechanism for slower rhythms. And interestingly, what we can see in these networks is that the network is quite flexible. You can switch between different states depending on external inputs, and that's also consistent with a lot of observations indicating that rhythms in the brain are highly transient. Then the network seems to be able to go in and out of rhythms on relatively fast timescales. I think I'm more or less, I should probably stop now. I had a few slides on what you can do with structured networks and things like attractor dynamics and things like that, but I think probably the later lectures will mention this, so I don't need to go into this.